Hello, grade 12 students. Here we start with our new chapter, Genetics. Chapter 2, Transmission of Genes and Genetic Recombination, Document 1, Hereditary Traits and Gene, pages 42 and 43 on your book. Chromosomes. Chromosomes, they are the carrier of the genetic information. At metaphase, they are made up of two chromatids, so they are distinguished easily. The substance that make up the chromosome is called chromatin. That's divided either into DNA or proteins. Proteins, they are composed of histones and non-histones, whereas DNA is composed of four nucleotides: A, adenine, T, thymine, C, cytosine, and G, guanine. A, T, C, and G, they are the four nucleotides that compose the DNA. A and T, they are complementary, whereas C and G, they are complementary. For example, if you have a sequence of DNA starting with the letter, for example, ATC, GCA, okay? First of all, every one of these is called a nucleotide. Every three together, they are called triplet, or we can call them codon. So every three, they are called codon. Later, every one of these will code for an amino acid. The complementary of A, it's in the DNA, as we know, it's double helix. So DNA double helix, DNA composed of nucleotides that are complementary. The complementary of A, it's T. The complementary of T, it's A. The complementary of C, it's G. Whereas the complementary of G, it's C. Complementary of C, it's G. Complementary of A, it's T. And this is the DNA double strand. DNA, by the process of transcription, they give mRNA. And mRNA, through the process of translation, they give polypeptide, or a sequence of amino acid that will go finally to a protein. So you have to know that the DNA, by the process of transcription, it can code for mRNA, messenger RNA, and then mRNA, by the process of translation, it will give polypeptide. What is a gene? A gene is a fragment of a DNA, it's a segment of DNA that code for a polypeptide coding for a trait. Alleles. What are the alleles? Alleles, they are different versions of the gene. For example, blood group gene. Blood group gene has three different alleles. It has the allele A, allele B, and allele O. Sure, you know that we have four different blood groups. Blood group A, blood group B, blood group AB, and blood group O. How can we have blood group AB? We'll talk about this in the coming slides. Homozygous or pure. What's the meaning of homozygous or pure? You have to know both terms, homozygous and pure, both of them. So individuals that have two identical alleles of the same gene. Let's talk here about the blood group. For example, an individual having two alleles that are identical. For example, allele A and allele A, 2A, AA, it's homozygous. Or BB, it's homozygous. Or OO, it's homozygous. While if you have, for example, AB, or you have A and O, or for example B and O, so you are classified as hybrid or as a heterozygous. So heterozygous or hybrid is an individual that possesses two different alleles for the same gene. It's called heterozygous, or it's called hybrid. While the individual that has two identical alleles for the same gene, we call them homozygous or pure. Here, dominant allele and recessive allele. What's the difference between the dominant and the recessive? Dominant allele is the allele that expresses itself in the case of both homozygous and heterozygous, meaning that it can be expressed in the case two strong alleles together, or one strong and one weak. The strong allele is the dominant allele, while the weak one is the recessive. Recessive allele, it cannot be expressed unless we have homozygous or pure. So it can't be expressed unless two weak alleles together. This is the strong allele, the dominant, while the recessive, it's the weak allele. So weak allele cannot be expressed unless we have two weak alleles together. Whereas that with strong, it can be expressed if you have strong and strong or strong and weak. Let's have an example here about the color of the eyes. Color of the eyes, here they gave us that the brown is dominant over the blue eye allele, which is recessive. So dominant, usually it's symbolized by a capital letter, while the recessive, it's usually symbolized by a small letter. Let's see here. Brown individual. Brown individual having, it can have BB, two strong alleles together, or it can have B capital, B small, strong allele and a weak allele, and the strong is going to be expressed. So here the strong is expressed in the case of heterozygous. Or blue eyes, it should be weak and weak, so recessive allele with another recessive allele. This is the case. Maternal and paternal, that is BB, so it will give a brown. B small, B capital, also it will give brown, since the B brown is the dominant. B capital, B small, also the brown is the dominant, so it gives a brown, while B small, it will give the blue eyes allele. This is the case where we have two individuals, two couple having brown eyes, and they gave birth to a blue eyes individual. This means that the blue allele, it was present in the parents that are heterozygous, but it was masked. And pay attention to this word, it was masked in the parents. While here, individual having brown, individual having blue. Blue is the recessive, so it should have two blue alleles. And here we have black and blue. So we have 50% they have brown and 50% they have blue, half of them. They're talking about the case of codominant. Codominant, the alleles that are both expressed in the heterozygous individual. For example, here's the crossing between a blue fish and a red fish. The result is a striated one having blue and red color, whereas the other one, for example, white and red, the crossing will be mixing white and red own color. So this is the case of codominant. Also, we can talk about the blood groups that we have explained before. The blood groups, we talk about blood group A and blood group B. So blood group AB is the case of codominance, where both blood group A and blood group B, both of them, they are expressed. Phenotype. What is the phenotype? Phenotype represents the sum of traits found in an individual. For example, genotype, sum of alleles and genetic makeup that form an individual. So some of the alleles correspond for genotype, whereas sum of traits correspond to the phenotype. So genotype always codes for the phenotype. True breeding lines. What the meaning of true breeding lines is a line of individuals where the descendants, they always have the same phenotype as the parents. So for example, we have individuals having blue eye color, all the descendants will have blue eye color. So this is called a true breeding line. Homogametic. What the meaning of homogametic? Individual that gives only one type of gametes. For example, the female ovum having XX, so it gives only one type of gametes. So it's homogametic, it gives only the X. Whereas heterogametic is an individual that gives more than one type of gametes. For example, the male. The male, we have X and we have Y. So we have X, Y. So it gives X and it gives Y. So it's called heterogametic. Homogametic, one type of gametes. Heterogametic gives more than one type of gametes. Plagueis type is a species whose males are heterogametic. Males, they are heterogametic. Example about this, humans. We have males, they are heterogametic, having X and Y. Whereas Apraxis type, species whose males are homogametic. What the meaning of this? Species whose males are homogametic, having, for example, ZZ, or females having ZW, different sex chromosomes, like chicken and butterfly. So males, whenever they are homogametic, and the females, they are hetero, having ZW, different sex chromosomes. Example about this, the chicken and the butterfly. So called about the Apraxis type. Hollandric gene. What the meaning of Hollandric gene? Is the gene that is carried by Y chromosome. So we call it a Hollandric gene. Whereas bidominant individual having two dominant phenotypes, for example, bidominant individual having two dominant phenotypes. Example, individual having blood group A, which is dominant, A is dominant, and normal regarding albinism, so it having the allele D. So it has two dominant phenotypes for the blood group and for albinism. So here we talk about the bidominant, whereas birecessive, it has two recessive phenotypes. For example, blood group O and the one coding for albinism, so it's called birecessive. Dihomozygote. What's the meaning of dihomozygote? Dihomozygote, individual homozygous for both traits. So individual that is homozygous for both traits is called dihomozygote. Whereas diheterozygous, individual that is heterozygous for both traits, it's called diheterozygous. So you have to differentiate between dihomozygous, diheterozygous, and bidominant, birecessive. Here, there's an experiment performed on mice to locate the genetic information. Where is the genetic information found, which determines the coat color, for example, in this case? So what do we have here? We have a true breeding black female mouse crossed with a male of the same genotype. Embryo have 16 cells, they are formed. The nucleus of one of these cells is extracted, and then it's injected into the fertilized ovum that is taken from where? It's taken from a true breeding gray female mouse crossed with a male with the same genotype. So first, let's repeat. We have a true breeding black female mouse crossed with a male of the same genotype. We have the cells, embryo of 16 cells is taken, nucleus of only one of these cells is extracted, and it's injected into the fertilized ovum that is taken from where? This ovum was taken from a true breeding gray female mouse crossed with a male of the same genotype. What happens next? We have the two pronuclei, they are extracted. So we have extraction of the two pronuclei. Then the obtained ovum is placed in vitro culture for 4 days. Then the obtained embryo is implanted into the uterine cavity of a white mouse. And this is a true breeding female white mouse, not fertilized. It's ready for only for gestation. So this is the case of foster mother. What was the result? The result was the birth of a black baby mouse. So you have to know how to start, starting like this, moving by order to list to this result. So this indicates that the black color of the baby mouse, it was coated by the information inherited with the nucleus of the embryonic cells, not the cytoplasm or the cell membrane from the this mouse, the true breeding gray female mouse, or not from the uterus, from the white mouse. So here the genetic information, they are inherited with the nucleus, not the cytoplasm nor the cell membrane, either not from the nutrition in the uterus, from the white mouse. So here we can deduce that the genetic information is located in the nucleus. Note here, if the descendants always have the same phenotype of the parents, So the line is called a true breeding, as we said before. Let's move now to part 2. Chapter 2, Document 2, Transmission of the Allelic Genes, pages 44 and 45 on your book. So here we have the crossing between a gray female mouse crossed with a white male mouse. So gray female mouse, they are pure, so we have only one type of gamete, which is the G, which represent 100%. Whereas the white, it represents W, which is 100%. The generation gives F1 its hybrid, having genotype GW. So the gray is the dominant and the white is the recessive. So 100% they are gray. Then we have F1 crossed with F1 in F2. F1 crossed with F1, this is called self-cross. Upon crossing F1 with F1. So F1 they are hybrid. So in the males and the females, in the males we have G represent 50% and we have W represent 50%. Whereas the G in the males, in the females, represent 50% also, and the W represent 50%. So this is called the table of cross. So G gives 50 times 50 divided by 100, so it gives 25%. G times W, it gives GW 25%. G times W, so it gives GW 25% and WW 25%. So here the final result will be what? We have here it's a gray. Also this one is gray since gray is dominant. And this one also is gray since gray is dominant and can be expressed in the case of homozygous and heterozygous. So here 75% of the descendants they have gray color. Whereas only this one 25% WW it gives the white. This is the recessive allele. It cannot be expressed unless in the case of homozygous. So here 25% they give white color. So after crossing a female gray mouse with a male white mouse, we get homozygous F1. Homozygous meaning that all of them they have the gray coat. 100% they have the gray coat. So the character gray color is dominant over the character of the white color, which is recessive. So here you have to know that the parents are pure. The first step to do is to give the symbols. You have to give the symbols since after crossing a gray female gray mouse with a white male mouse, all the descendants they are gray. So gray dominates over the white. So gray is dominant. So we have to give the symbol. Let G capital be the symbol of the allele coding for the gray coat. And let W small be the symbol of the allele coding for the white coat. So here W small since it's recessive. In F2, what is F2? F2 it's crossing F1 crossed with F1. So F1 crossed with F1, what do we call this? It's called self-cross. In F2, we get mice with the white coat, whose genotype is WW. White is recessive. So they should inherit the allele W from each of the parents of the F1. That should be heterozygous, having genotype GW. Homozygous or heterozygous, always it's used for the genotype. Whereas homogeneous, heterogeneous, it's used for the phenotype. Factorial analysis. Crossing the gray with the white, GG. Parents, you have to put, for example, first the phenotype of the parents. Then you move second to the genotype of the parents, GG crossed with WW, since the parents, they are pure. Number 3, you have to move to the gametes, 100% G, and here 100% W. Number 4, genotype of F1, it gives 100% gray color, since the gray is dominant. And number 5, phenotype, mice will have gray coat. F1 crossed with F1, so we have GW is crossed with GW, so we have 50% gray, 50% W, since we have two types of gametes, 50% and 50%. And here we have G 50% and W 50%. So the table of cross will be GG 25%, GW 25%, GW 25%, and WW 25%. So the final result will be, as we said before, 75. This will give gray. Also, this will give gray. And also, this will give gray. So 25 plus 25, 50 plus 25. So it will give 75% that you have gray, and only 25% that have. So 25% GG, 50% they have GW, and 25% they have WW. So the phenotype of the F2, 75% mice of gray coat and 25% of mice with white coat. So the results, they are verified according to the document. Notes: tested cross. What is the tested cross? Tested cross is the cross that is done between an organism whose genotype for a certain trait is unknown. We don't know the genotype of the individual, and an organism that is homozygous recessive. This one, the known one, is homozygous recessive. So the unknown genotype can be determined by this cross. For example, we have this unknown dominant phenotype, either B capital or B capital B small. We cross it with homozygous recessive, B small, B small. If the unknown is B capital, so we will get only one phenotype. 100% they will be black. While if the unknown is heterozygous, we will have 50-50. So 50-50, the unknown will be heterozygous. 100%, the unknown will be homozygous. Codominance and intermediate dominance. Some cases, both alleles they are independently expressed. For example, in the case of the blood group, alleles A and B, both of them they are dominant. So in the case of blood group A and B, they are dominant. If they are both of them they are expressed together, here we are talking about codominance, and this occurs in the individual having blood group AB. So AB, it's the case of codominance. Okay, codominance where both alleles they are dominant and both of them they are expressed. Other cases, dominance is incomplete. So we have incomplete dominance. How? Incomplete dominance means that we have the case of intermediate dominance. For example, in snapdragon plants, a cross performed between two breeding lines, one having red flower and the other having white flower. Crosses and the results they are obtained in this document, document E. So what will happen and what is the result, and how can we determine that this is the case of incomplete dominance? Here we have red flowers, okay, and the parents here they are pure. So red, they are R crossed with the white, WW. Here they give only one type of gamete, which is the R. And also here they give only one type of gamete, which is W. If you notice here, both of them they are dominant. And the pink, we have RW. So RW, pink flower. Pink is intermediate between red and white. So this is called intermediate dominance or incomplete dominance. Okay? F2, upon crossing pink flower with pink flower, means that crossing RW with RW. So we have 25% having red, 50% having RW, and 25% having WW, which is the white. So codominance, both alleles they are expressed. Remember, in the case of, for example, the case of the red and the blue fish, the color will be fish striated with blue striated with red, or red striated with the blue. This is the case of codominance. Or like the case of the blood group, where both of them they are expressed in the case of blood group AB. While if we have an intermediate color or incomplete dominance, like the case of red flower crossed with the white, the color will be pink. So it's intermediate between the red and the white. So this is the code intermediate. Both of them they are dominant. Here RR, both of them they are capitalized. So both of them they are dominant. This is everything related to the first two documents of the genetics chapter. Thank you.